Assalamu alaikum students. Before going to our educational topic, please subscribe my channel Muntzir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic. Today we will talk about the oxidative phosphorylation. That what is oxidative phosphorylation and when and at which points does the oxidative phosphorylation occur. First we see the definition of oxidative phosphorylation. Students, oxidative phosphorylation is the formation of ATP, synthesis of ATP from inorganic phosphate and ADP, meaning that by the adding up of inorganic phosphate and ADP, when the transposition of electrons, transfer of electrons occur from the donor molecule to an acceptor molecule, when there is happening electron transport chain. So students, oxidative phosphorylation is the synthesis of ATP, production of ATP when inorganic phosphate combines with adenosine diphosphate, which results in ATP. In simple words, we can also say that the generation of ATP, the production of ATP molecule in the presence of oxygen is called oxidative phosphorylation. Students, oxidative phosphorylation takes place during respiratory chain, during electron transport chain, and 3 ATP molecules are produced during the 3 steps of the electron transport chain. As you can see here in this electron transport chain diagram, that there are 3 points in this electron transport chain diagram during which 3 ATP molecules are produced. First ATP molecule is produced when NADH2 molecule is oxidized. Here in between NADH2 molecule and in coenzyme Q. Second ATP molecule is obtained when electrons discharge from the cytochrome B to cytochrome C. Here at this diagram you can also see that when electrons flow from cytochrome B to cytochrome C, second ATP molecule is prepared. A third ATP molecule is synthesized when electrons shift from cytochrome A3 to the molecular oxygen. Here at this point, you can also prove that when these electrons shift from cytochrome A3 to the molecular oxygen, then this third ATP molecule is synthesized. All these express that and help us to understand that when a substrate molecule is oxidized to NADine by an NADH2 molecule coupled by the enzyme dehydrogenase, 3 ATP molecules are synthesized for every atom of oxygen that is reduced to form one molecule of water that is finally produced here at this point. Students, all this electron transport chain happens for every NADH2 molecule that is obtained from glycolysis or from Krebs cycle. In another case of succinate oxidation during the Krebs cycle, when succinyl coenzyme A is converted to succinate, 2 ATP molecules are synthesized for every oxygen atom. Here from this Krebs cycle, you can watch that 2 GTP molecules are produced by the twice repetition of Krebs cycle. These 2 GTP molecules are equivalent to the 2 ATP molecules. Students, before watching this video of oxidative phosphorylation, you must watch the videos of glycolysis, Krebs cycle and electron transport chain. In this way, you can truly understand the ATP synthesis. Students, when you will watch the video of electron transport chain, the first part, then you will completely understand the movement of electrons from NADH2 to molecular oxygen. It will also help you to understand the mechanism of oxidative phosphorylation. Students, the oxidative phosphorylation can also be expressed by this following equation. When NADH reacts with hydrogen ion in the presence of 3 molecules of ADP and 3 molecules of inorganic phosphate in the presence of molecular oxygen. Then it will help us to obtain NADine, that is nicotinamide adenine dinucleotide ion, along with water and 3 ATP molecules. Here this pi is showing an inorganic phosphate. This molecular mechanism of the oxidative phosphorylation is associated with the electron transport chain, that is the respiratory chain. This respiratory chain is present in the inner mitochondrial membrane. The mechanism of oxidative phosphorylation is chemiosmosis. Therefore, next we will understand the chemiosmosis. Students, chemiosmosis is a coupling reaction during which the synthesis of ATP molecule occur during the movement of hydrogen ions across an hydrogen ions gradient. Therefore, the definition of chemiosmosis is as the coupling reaction in which the synthesis of ATP molecule takes place during the movement of hydrogen ions across the hydrogen ions gradient is called chemiosmosis. Chemiosmosis is a special process which utilizes the mitochondrial membranes during the redox reactions for the ATP production. Students, although substrate level phosphorylation is an oldest method for ATP production, but more and more ATP is generated from an other process which is called chemiosmosis. Now we will see the mechanism of chemiosmosis. Students, eukaryotic organisms possess transmembrane channels in their mitochondrial membranes that pump protons. Suppose we have a mitochondria. If we see this small portion of a mitochondria under a microscope, then we will obtain this view. In this view, it is the outer compartment of mitochondria, whereas it is the inner compartment of mitochondria. This mitochondrial membrane possesses an enzyme which is known as ATP synthase, and this mitochondrial membrane also consists of a proton pump. As we know that glycolysis, Krebs cycle and beta oxidation produce reducing equivalents which include NADH and FADH. NADH, that is nicotinamide adenine dinucleotide, and FADH, that is flavin adenine dinucleotide, which possess electrons. These electrons are transferred to the set of enzyme complexes. I have already prepared a video of enzyme complexes of the electron transport chain. If you want to watch this video, then you can. When electrons travel from first enzyme complex towards the fourth enzyme complex, finally during this movement of electrons from the first enzyme complex towards the fourth enzyme complex, energy is generated. This energy is utilized in pumping the hydrogen ions from the mitochondrial matrix into the intermembrane space. Here is present mitochondrial matrix, whereas it is the intermembrane space. When this NADH reduced form donates its proton, then it attains a oxidized form, that is NADine, and this proton travels from this proton pump and enters into the intermembrane space. This continuous pumping of hydrogen ions from the matrix to the intermembrane space causes the generation of higher concentration of hydrogen ions in the intermembrane space as compared to the matrix of the mitochondria. This movement creates a positive charge in the intermembrane space and the negative charge in the mitochondrial matrix, which creates an electrochemical gradient. As these hydrogen ions cannot cross the electrochemical gradient because the inner mitochondrial membrane is impermeable to hydrogen ions. So here we need a specific transporter, a specific vehicle, that is called the enzyme ATP synthase. At this point, when the concentration of hydrogen ions become greater inside the intermembrane space as compared to the mitochondrial matrix. So here an enzyme ATP synthase comes for its proper functioning. This enzyme ATP synthase transports hydrogen ions into the mitochondrial matrix from this intermembrane space and utilize the energy generated from the flow of hydrogen ions, movement of hydrogen ions to phosphorylate to combine adenosine diphosphate, that is shortly called ADP, to adenosine triphosphate, shortly called ATP. This is the basic concept of energy generation in the cells in the form of ATP, which is also an energy currency of cells in any organism's body. Students, these are the redox reactions which occur in the enzyme complexes of the electron transport chain, which are present in the inner mitochondrial membrane, causes the pumping of hydrogen ions. These hydrogen ions from the mitochondrial matrix into the intermembrane space. These hydrogen ions are obtained from NADH2 and FADH2. When this pumping happens again and again and again, then all this pumping of these hydrogen ions into the intermembrane space causes the generation of higher concentration of hydrogen ions into the intermembrane space as compared to the lower concentration of hydrogen ions inside the mitochondrial matrix. As we can see that here is higher concentration of hydrogen ions into the intermembrane space, and here is lower concentration of hydrogen ions inside the mitochondrial matrix. As intermitochondrial membrane is impermeable to hydrogen ions, therefore these hydrogen ions cannot cross this impermeable membrane of mitochondria. As such here, therefore here happens the function of an enzyme which is known as the ATP synthase. This ATP synthase enzyme is present in the inner mitochondrial membrane and it utilizes the flow of these hydrogen ions, the movement of these hydrogen ions from the intermembrane space that has higher concentration of hydrogen ions to the mitochondrial matrix that consists of the lower concentration of hydrogen ions to produce enough amount of energy. This energy is used to phosphorylate the adenosine diphosphate, shortly called ADP, to adenosine triphosphate, that is called ATP. This is the basis of ATP energy generation inside the any organism's body, and this is the whole mechanism of chemiosmosis which occur inside the inner mitochondrial membrane via an enzyme ATP synthase and a proton pump, that is a protein. Students, these proton pumps utilize a flow of electrons to induce a shape change in this protein. Therefore, it causes these protons to shift out to flow out the inner compartment of mitochondria, as from mitochondrial matrix into the intermembrane space, and then rest of the phenomena happens. Students, how you find this lecture? Please tell me by liking this video and subscribing our channel Muntzir Mehdi Science TV. I hope this video will be helpful to you to understand the mechanism of chemiosmosis and oxidative phosphorylation, because same this is the oxidative phosphorylation when ADP combines with this inorganic phosphate to synthesize this ATP molecule in the presence of oxygen during electron transport chain. This same concept is the concept of oxidative phosphorylation. So students, at the end I will encourage you that you have no need to chime this topic, but consider it, understand it with the help of its diagram. When you will understand all this mechanism of chemiosmosis and oxidative phosphorylation with the help of its diagram, then you can write this mechanism of chemiosmosis and oxidative phosphorylation in your own words. Ultimately it will become very easier to you, as chemiosmosis is an important topic that often comes during annual examination. Lastly, thank you for watching this your video.